welcome once again, everyone, to this Interact online lesson. And in this one, we will talk a bit about soil and what we researchers do with soil and why we are interested in it, and also what, in the end, what you can do actually to help soil health. And the title suggests really that it's a bit of an unknown microcosmos that we have actually not a lot of knowledge about. And you will see why and what we are trying to improve there. My name is Cornelia Klütsch. Before we really start, I thought I'll just talk a tiny bit more about myself. I'm a biologist and I work as a researcher on a biological station called EDU-ARCTIC. So here at the left-hand side, there's also a picture from the summer of the station with a little pond in front. Right now it's quite dark because we are 400 kilometers north of the Arctic Circle. And right now the sun does not come over the horizon. So it's sort of the dark time in the winter for 6 to 8 weeks. The station is located basically at the most northeastern tip of Norway, very close to the Russian and Finnish borders, in this little wedge here. And it's actually very, very close to the Russian border. So the station is here, and you can basically walk sort of probably 15 minutes to a river called the Pacific River, which I talk about quite a bit in other presentations for Interact. And the middle of the river is basically the border to Russia. So it's a shared border between Norway and Russia, and our Russian friends are over here. We have quite a number of Russian colleagues we are working together with. And because we are situated so far in the north, a lot of research we are doing deals with changes like climate change, but also pollution and quite a bit of ecological research going on. A lot of research, however, is also connected to genetic methods, and we have a genetic lab up here. In the past, we mainly studied large mammal species like brown bear and polar bear, but over the last sort of 5-6 years, we extended our research focus and study now fish species, for example, in the Pacific River, but also other organisms like insects. And it's connected to this presentation. We have recently started looking more into soil organisms. And I will explain why in a minute. But before I do, I just thought I'd give you a short overview what we will cover today. And the first question is actually to give you a bit of introduction and a few explanations and definitions, and a bit background why we actually study soil. What is known, what is not known currently, what will we do in the future. And then we will have a sort of sneak peek into one of the projects we currently have at EDU-ARCTIC. And a bit on what methods we use. And I specifically put a focus on here which methods can actually be used, for example, in classrooms as a class project as well. Then we have ecological functions. I will introduce you to a few soil groups. There are many, and I won't have time to cover them all, but a few key groups that we are studying and why, and what their tasks are actually in soil, why they are important. And I will close this talk basically with a bit on what every one of us as citizens can do to improve soil health and why this is important and how this is connected to climate change actually. And finally, as said, if there's time, there will be a Kahoot. It depends also a bit on how many people we are. So we'll see if there are enough people to do this. So first of all, it's very important to realize that soil biota, so all the species that we have in soil, are equal to 25% of global biodiversity. So all the species we have, all the biodiversity that's around us, 25% is found in soil. That's quite a striking number because I think a lot of people are not aware that a quarter of all the biodiversity is found in soil. Because we probably humans are probably more drawn to species that they can see and that are not in soil but above ground, like trees, bears, and other species. So if we can't see it, we tend to forget about it. But particularly in soil, it's very important that we take care of the soil biodiversity. I will talk a bit more about this later. So we know actually not a lot about soil species, the biodiversity, and the ecological functions they have. So there's a real research need to do more studies on soil and what lives in soil and how this all works. And this is particularly important because we know quite a bit about above ground diversity. As I mentioned, we know quite a bit about the plants that we see. But in soil, and the soil organisms are very much important and connected to, for example, plants, because a lot of soil organisms live between the roots of plants and have important functions for plant growth, for example. And from a research side, this goes back to a lack of coordination. It seems that there are just sort of isolated studies, and we as a research community have to become better, to be more strategic how we summarize all the knowledge that's there and identify also more strategically which studies need to be done. And just to drive this point home in another way, how important and how much is in soil to be found, if you have just one hand of soil, as shown here, you have an incredible biodiversity. So you have something like 50 kilometers of mycelium. This is basically fungi that build a network. So that's quite a distance, 50 kilometers. 10,000 nematodes is one of the species groups we'll talk about a bit more today. If you think about bacteria, you have probably something like more than 100 million bacteria just in a handful of soil. Probably up to 5,000 insects, spiders, little snails and worms. Even algae, you have hundreds of species. And then you have in the 100,000s of protozoan cells, for example, amoeba and other species, and quite large biodiversity there as well. So if in a handful of soil, there's about 0.5 grams of living matter of these species, then this adds up to 5 tons per hectare of land. So the organic matter in terms of animal life and bacterial life in a handful of soil is quite tremendous. So it's really important that we take a closer look. And I mentioned a few terms here, and I just thought I repeat a tiny bit and also talk at the same time about the classification systems that I use, because we have this tremendous biodiversity with very diverse taxonomical groups. Actually, a common classification system is simply by body size. And we're starting with very small organisms here that are smaller than 0.1 millimeters. This is mainly bacteria and other microbes. Then we have microfauna. Micro is also very small, means small, that are similarly just slightly larger. And there we have, for example, nematodes and tardigrades. Tardigrades are called also water bears. And then mesofauna. Meso means something in the middle, so medium-sized. But as you can see, we are talking about small organisms, so they are hard to study also because they're so small, and they're usually below 2 millimeters. And there we have a lot of mites and springtails and a few other groups. Then we have macrofauna. Macro means large, but large means in the soil world above 2 millimeters. And there we have ants and termites and isopos and earthworms and some beetles and so on. And finally, we have megafauna. It's very large. But here we meet basically frogs and some mice and voles and so on. And what is quite interesting is that with increasing body size, the abundance is quite low. So if you have increasing abundance, the smaller the organisms are. So we have usually very high numbers of bacteria, but then still fairly high numbers of some of those mites and springtails, for example. But in comparison, the number of mice, for example, is quite small, because we are talking here about millions and here probably about thousands or less, depending on the area you're looking at. So this is a common classification system, and just to introduce you to a few more terms that we are using. So I talked a bit about that we don't know so much, and what people have actually tried to do, researchers have tried to summarize actually the knowledge, what has been done. And one group focused here on mapping existing studies onto a world map. And they wanted simply to know what has been done and where in the world. They focused on 3 groups that are listed here. So the different colored circles means different groups. In blue, this is fungi. In yellow, we have bacteria. And in red, we have macrofauna. So remember, macrofauna is, for example, earthworms, ants, and such critters. What we see in the distribution here is that particularly in North America and Canada, Scandinavia, and then Russia and Siberia, not so much has been done. This is partially probably because we have more ice cover there, but with climate change, some of these ice covers will disappear, and we want to know what's happening there more. But also it means that, for example, even in Scandinavia, where there are non-permanent ice covers really, basically only part of the groups have been studied, in this case soil fungi, but a lot of the other groups seem to have not been studied. And this is quite interesting. It represents really a picture where we need to do as researchers more to understand more about soil. What this also shows, I told you in the beginning that some of these other species like mites and springtails, like these mesofauna, these intermediate size specimens, they seem to be not even listed here. My suspicion is that there not good databases exist for those type of specimens, and therefore they could not be plotted, again, reiterating that we need better databases. Then another study looked more at the sample effort. So these circles that are plotted on this world map again, the larger they are, the studies they looked at used more sampling sizes. So these larger, bigger circles have more than 100 sampling locations. And again, you get a similar picture. Europe, including Scandinavia, quite a bit has been sampled, but in general, the North is probably a bit understudied or undersampled. But this becomes even clearer if we look here at the graph at the bottom, that the sampling effort for different groups is very different. So there are far more studies that look at bacteria and ants, for example, than at mites and springtails. And finally, I thought I show a picture here that again deals with biodiversity of different groups, but here is shown the proportion in this greenish color of the species we know already. And in this dark blue, the proportion of species that we suspect exist in nature, but that are not studied or described yet. They are unknown to science and the world. What we see here is that some of these species, like mammals, but also fish species and larger plants at least, have quite a good proportion of species that are already described, so we know about them. However, even if we go to insects like flies, butterflies, and bees, so really species that we can still see with our own eyes, but are fairly small, we see already that the proportion changes quite a bit. So there's a higher proportion of species that we don't know about yet. We suspect that they exist, but science has not had the opportunity to describe them yet or to learn about them. And finally, if we go into the soil, we see that the proportion of unknown species is very high. So for nematodes, only 2% of species diversity is probably described. For springtails, it's a bit more, but still a high proportion unknown. Even for earthworms that are, at least in the soil world, quite large species, they're still probably 75% of the species are unknown to science. Why is this important? If we don't know about them, we can't protect them, but we also don't know what they do actually in the soil, which would be quite important to know if we want to improve soil health, which is one of the sort of end goals we have with this research. And below here, I just give a few numbers. These numbers change a bit depending on the source. Just to give you an example that really in mites, for example, we have described probably, if these numbers are a bit older, probably 60,000 species are described, but we suspect that there are about 1 million species out globally. The picture is pretty similar for these groups. Springtails a bit better. But as you can see, just by these numbers, we have a lot to do and a lot to learn. And this is why we here at this biological station set up a project to study in more detail 3 of these understudied groups, which are springtails, mites, and nematodes. And this is simply because we saw from the literature that this represents a real knowledge gap, so not a lot is known. And particularly, not a lot is known in the subarctic or in the northern regions. And part of this project is always to have a lot of collaboration partners. So these are listed here. And the reason for this is because it takes a lot of time to become actually an expert in each of these groups. So we need an expert for mites that really can take a specimen and look at it under the microscope and find out, okay, this is probably this species, but this looks new, this looks different. Maybe this is a new species. And to describe this biodiversity, we need basically experts that have really learned to determine those species and to get to know these species over 10 or 20 years. It takes a long time to train up a person that is a specialist in a particular group, like mites or springtails or nematodes. And then on the right-hand side here, I just show you some of the sampling locations that we have along the borders here and further in the north. And the idea is simply to cover as much ground as we can, different habitats. For example, birch forest and pine forest, or more lake shores and other soil types, because we suspect specialists in each of these habitats, so different species that have different, are differently adapted to certain habitats. And I will talk a bit later about this following part. So these experts do this with microscopes. So they're looking at the specimen, the morphology. But we can do this also, and this has become more popular in the recent past with genetics. So part of this project is also to come up with basically a barcode, like you have a product barcode on a product that you buy. We can use genetic information to create a barcode that we can say, okay, this piece of genetic information is unique to a particular species. So we combine these 2 methods to then be able to later, with the genetic method, to identify species more quickly. I will talk a bit more about this in a minute. And with this, I have sort of mentioned also already a bit the methods. I thought I'd give you a quick overview of methods we are using and how you, for example, can use those in a classroom setting if you're interested to also study a bit of soil biodiversity. So we'll talk a bit about collection methods and then modern methods, which mainly the genetic methods, and then one ecological methods that can also be used to study soil activity and soil health. Starting with some of these collection methods, basically what we are doing is we go out in the field. And again, we have probably a study design that we say, okay, we want to collect in different habitats, like different forest types or more tundra habitat. And then we collect samples, take photos of the vegetation that is found around the sample, as this may give us some more background information about the spot. And here, for example, we always have an identifier and a date so that we can really track the samples we have taken. And another method is to collect, for example, moss material, like litter material, because some of these soil organisms are also found in that part of the habitat. And here you see one of those taxonomic experts, Arne Fjellberg, collecting some of that material and looking at it if, for example, some springtails are in that material. More generally, what we are using is a device called a Berle's funnel or Berle's trap, where we put the soil sample on a mesh. So we have a soil sample here with our little critters. Then we have and a funnel. And then on top we have a light bulb, which of course gives light, but also creates heat. And one of the characteristics of soil organisms is that they don't like light, they don't like heat, they don't like dryness. They like it moist and dark. And what this light bulb does, it dries out sort of the surface of the soil. And of course, the surface will be also warmer than sort of the bottom of the soil sample here on the mesh. And these specimens migrate towards the bottom, and at some point they fall through this mesh into a collection jar. And in this collection jar, we can take them and look at them under the microscopes and study them. So, and then really the experts look at them one by one to see if there's anything new in there, or if it's new probably to Norway or new to science. And here, this picture or the slide I just put in there, although that we have custom-made Berle's traps, but I put a link here to a journal that's specifically designed to give scientific information to kids. And it's quite interesting because one article that I have linked here describes how you can make your own Berle's traps for yourself privately or as a school project. And as you can see, the material you use here is quite everyday material: a light bulb, some cardboard, scissors, some tape, sort of a sieve as a mesh. And then it's exactly described how you can set this up to collect your own specimens as a little class project, for example. Then I mentioned nematodes. There we have a similar but slightly different method, which is called the Behrman funnel. And the Behrman and Berle's, these were scientists that invented these methods, by the way. And this is also fairly simple, actually, that you have, again, a funnel, but this time with a silicone tube and you have sort of a squeeze clip, because you have water in this funnel and hose. And then again, you have a mesh and sort of a plastic PVC tube. And you put your soil sample in here, and nematodes tend to, at some point, go deeper. They like it moist, go deeper into the soil. And then they're basically collected at the bottom. They sink to the bottom here, and then we can just release a bit of water and those collected nematodes to then collect them in a collection jar and again look at them under a microscope. However, I sort of hinted already that this is a fairly slow method. So you have to go out in the field, collect your sample. Like the Balaise extraction, for example, depending on the moisture level of the soil sample, we leave them for 10 to 14 days. And after this, the experts look at them, and really you need an expert for every group, and you need an identification one at a time, which is quite time consuming. And because we are basically losing biodiversity right now because of climate change and habitat degradation, we scientists look for ways to speed up the process to find actually all these species and describe most of them if possible. And what has been done? Well, there are some other problems. One of them is that some species are so similar that we actually can't just rely on morphology, just on the exterior appearance alone. For microorganisms, about 90% of the species actually can't be cultured in a lab. This means we can't grow them, and this means we can't study them in detail. And what this means is that with this approach, we basically rarely come to study more like ecosystem roles these species have, and it's quite slow. And therefore, in recent years, DNA-based methods, so using genetic methods, have been developed. And I just run you through this experiment here. But if you want to know more about this, I have also one or two other talks in this webinar series where I talk a bit more about the genetics and the genetic background of these methods. In this case, we have our soil sample, and we are able to take a tiny bit of soil and extract DNA from it in the lab, and then have other lab procedures that in the end give us a sequence, a series of nucleotides. So DNA has 4 nucleotides. These are 4 different colors here. And this is A, T, C, and G, standing for 4 different nucleotides. And a series of nucleotides we can compare between samples. Let's say we have 2 samples, and I've highlighted here in red differences in those sequences. And by comparing this, we probably come to the conclusion, okay, so they are so different from their genetics that they are most likely different species. And the main advantage of this is that it's faster and we can study more species at the same time, simultaneously from a habitat. So these two approaches together are quite the morphological and this genetic-based method together gives us a very good indication what is in the soil sample we are looking at. And finally, I thought I'd talk a bit about an ecological study. And what has been used quite a bit are so-called bait Lamina strips. These are these strips that are shown here at the bottom. These are basically plastic strips usually, and they have holes in them. And those holes are filled with quite some yummy food that soil organisms like to eat. And they're basically put into the soil in usually there's a study design in the background. In this particular example, it was sort of a controlled site. It did not have pollution and a polluted site. And the scientists wanted to know, okay, does pollution actually affect activity of soil organisms? Because this would, of course, be an indication that they're not doing well or that the level of biodiversity is quite low. So they put out these lamina strips in the soil of these 2 habitats and compared how many holes are actually either empty or half eaten, and sort of summed this up and could then give an indication. And you probably can see just from looking at it that you have really empty holes in this controlled site, but the polluted site, only a bit of activity was found, indicating that this polluted site has probably quite a lower level of species and activity going on. And so that's something, this pollution may have a strong impact on the soil health in total. And the middle part, yeah, so I studied also how to analyze these soil strips, because originally you only said there is something eaten and not, and this was a pretty rough scale, whereas they came up with a more refined scaling scheme that they say, okay, it's fully eaten or half eaten or just a bit eaten, and then if you take this into account, you just have a bit of better resolution in your interpretation. So this is just this middle part. So generally we can use those to see if there is a difference in activity of soil organisms, and then we can study, for example, polluted versus non-polluted sites or different habitats, for example. So this gives us also an indirect indication of soil biodiversity. Okay, so we have talked a bit about different methods we are using, and that can be also used by classrooms, for example, to set up a little experiment. And so now I thought I'd talk a bit about one or two groups in a bit more detail what functions they actually have. These are just examples. So there's far more biodiversity out there. But I thought I'd talk about mites and springtails because these are some of the groups we are studying. Starting with soil mites, we have soil mites are a quite successful group. They're found almost everywhere in soil. And they have three major groups. These are these three pictures. It's not important that you remember the names, but these little pictures are also found below here again and indicate simply which groups have which function in the soil. And if we start here with the plant feeders, a lot of people, I think about soil when they think about soil organisms, think about that they're probably eating sort of roots and parasites. But for example, for the mites, this is absolutely not true. There are very, very few species that are actually connected and eat live plant material, living plant material. Quite to the contrary, they help in making and in recycling dead litter materials like dead leaves or dead body parts of other insects, and help therefore in the decomposition process that's happening in the soil to recycle this organic material and release nutrients into the soil, and therefore are also contributing to sort of a natural recycling and fertilization process in the soil. Then you have microbial feeders. So these are, you have also a food web in the soil. So some mites feed on microbial bacteria, for example. And by doing this, because they're grazing on them, they're also distributing them throughout the soil, for example. And by eating them, also there again, there's a recycling process in a way going on, releasing nutrients, and by dispersing them and so on, they help also with microbial growth and activity. And finally, we have a group that are predators that feed on other soil organisms like springtails or nematodes. And this is also an important function because what mites do, for example, we have nematodes, we have quite a number of species that actually are parasites and live around plant roots. And if we have healthy soils, then mites contribute by eating nematodes to reduce the population size of those parasitic nematodes. Just one example is giving here that sort of one adult mite and the offspring can consume about 20,000 nematodes in just 10 days. So they're quite efficient regulators in the soil and regulate the population size of other species. So this is just an example of ecological functions some of these species have. And as a second, and I just wanted to mention here that plant feeders are called also herbivores, yeah, just as a additional piece of info. And then we have springtails as a second group. And here on the left-hand side, Just a few pictures, how they look under the microscope, close up. And here's sort of a schematic outline. And the most important thing here is that they have an appendix, which is called a forker to jump. So springing, so jumping. This is really the preferred way of transportation for springtails. And just to show you this a bit more, I have a video and I hope this actually works. Just wait a tiny bit. Yes, so just a little impression how springtails actually move if we find them sort of above ground, but they live also, a lot of species will live below ground in the soil. And that's just as a second example, I thought that I also talk you through some of the functions they have. It's quite similar actually to the mites, so it's a bit of repetition also, which is probably good. And if we start here, then also springtails contribute to basically a natural recycling process of organic material, as maybe dead leaves, for example. And this process is also called decomposition. And so they help also by this decomposition process, they help to form soil actually. And this is quite important that these soil organisms contribute thereby also to soil structure. So how the soil is built up. Some soil organisms, like for example earthworms, have also the function to, because we know they have these little tunnels in the earth, they contribute also quite a lot to the soil structure, and that we have ventilation and also water transport, that the water can easily get into the soil. So springtails also contribute as one group to soil structure built up. Then we have, they're also feeding again on microbes and help particularly around the roots, that microbes are transported and dispersed. Then we have some springtails are found around the roots, and because they help with this recycling process, they also get nutrients closer to the roots of these plant species and help thereby with nutrient supply. And then the springtails themselves can be food for, let's say, spiders or other animals. Again, this is a food web function, so that also other animals find food. And then there's some springtails that live on plants, and there they're feeding also often basically of microbes, of bacteria, and help with dispersal. So again, these little creatures have quite a versatile set of functions in the ecosystem, and they are very important to improve physical and chemical properties in the soil. And really, they're all connected. It's a network that is very important to be intact. So more general and just summarizes a tiny bit that a lot of these soil organisms, we haven't talked about all of them, but every little critter in soil has its tasks to contribute to overall soil health, to help with recycling, decomposition of organic matter, and also regulates population size of other species. And it's important to get nutrients to plants, for example, which of course is connected, for example, also to us humans in food production. And they contribute by releasing these nutrients, they contribute to different nutrient cycles like nitrogen, carbon cycle, and water cycle. And by contributing to the soil structure, they improve also water filtering, so taking contaminants out of the water so that we have good ground water, which we partially use as drinking water. And by releasing this, they're very important in a natural fertilization process. So we humans have used quite a bit of synthetic fertilizer, and I will talk in a bit why this is not a good idea. So an attacked ecosystem, soil ecosystem, means that it's a quite good, healthy and nutritious soil that of course lead to more nutrient-rich plants and also better growth of plants. In the very beginning, I talked a tiny bit about that climate change is quite important for us here in the north because it's happening faster. So the temperature changes are faster here than in more southern regions. So I thought I quickly give you one example what we might expect in the future. It's important to realize that just very few studies have actually looked at it. So it's another area where we need to do more research. But starting with two parameters, it's temperature and precipitation, so rainfall. And these are important for soil organisms because, as I said, they don't like it hot, they don't like it dry. So they want moisture and darkness. That's why these two factors are drivers in annual changes. And sort of these cyclic annual changes we see in either occurrence or in different life stages is also called phenology. And these changes over the year, they are timed quite accurately by nature. And if there are mismatches in different processes, then often the system doesn't work properly anymore. And this is one of the main concerns we have with climate change. It's distributions of species change or activity levels so that some of these ecological processes do not fit anymore. There are mismatches in the timing, which can be a problem. And if we first start with this picture here, we have a location. It's a hypothetical location where we have in red the temperature over the year, and the bars are then the rainfall amount over the year. And as you can see in this particular location, we have currently a climate where it's hotter in the summer, cooler in the winter, and you have probably the most precipitation somewhere in autumn, but overall you have some precipitation throughout the year. And here we have some of those soil organisms I have been talking about. And what's important to realize is that most of those soil organisms are found probably in the top 10 to 15 centimeters, which we call also the topsoil. It's at the top in the soil. And here most of the activity pattern happens. If this is a graph showing activity according to depth in the soil. So in the topsoil, just below the surface, we have most of the activity because we find most of those organisms in that area. And as I said, spiders or beetles feed on some of those soil organisms, and these soil organisms are important for nutrients so that the plants can grow well. And because we have a higher temperature in the summer, we have over the year, this is again time and this is activity, we have sort of a dip in activity usually in the summer because it's quite hot. And again, the soil organisms don't like this. What might happen with climate change, it's first of all that weather events become more unpredictable, but probably in general, we have an increase in temperature. This is one of the predictions. And in this particular example, we have also less precipitation suddenly in the summer. So it's hotter and drier in the summer. And what happens is that then generally these soil organisms will migrate further into the soil. Similar to the Berlais funnel trap I showed, because they don't like heat, dryness, and they will go down where it's cooler and where there's more moisture. So the activity level, the spatial activity level will be shifting. You won't have necessarily most of the activity directly in the topsoil layer because these animals are migrating lower. This can create a problem with this nutrient cycling I talked about. So because of less activity in the topsoil, those plants will probably get less nutrients, for example. And because these specimens go further into the soil, also some predators like beetles may have body size shifts because they can't find enough springtails to eat, for example. So again, there's a cascade effect of different things, and you probably don't have as good of plant growth either because of the lack of nutrients. And finally, if you look at this graph, the activity level will be very, very low in the summer because it's dry and hot at this point. So we may see quite a lot of changes in soil when it comes to climate change. And we have to think about, if we think about food security for humans, for example, we have to think about how we can combat this and come up with strategies that increases soil health. So if you look here first at the right-hand side, it just summarizes a bit what happens, what I have mentioned. So in the past and still ongoing currently, we have quite intensified agricultural practices where we use a lot of fertilizers, synthetic fertilizers. We have climate change happening in the background, but we may have also other pollution like industrial pollution going on. And all these factors lead to that these soil organisms decrease in abundance. And so we have generally low biodiversity in soils if it's very intensified agriculture. And this means that a lot of these natural processes are not functioning anymore very efficiently, but also it means that other species that are pests or parasites, that they can increase in number. Remember that I talked about this with the mites, that they regulate the number of specimens of nematodes, parasitic nematodes, for example. And what is increasingly done, and what a lot of research goes also into, is how do we do this better, especially because we have a changing climate. So the aim is, of course, to have high biodiversity, a high number of these beneficial organisms that help with soil structure and water regulation and so on, and that we have by this also a lower number of pest species, so also a lower use of pesticides, for example, that are also not very good for nature, but also not good for humans. So it's really sort of, again, everything is connected. If we have good animal health in the soil, then we have better plant health, and by this also humans benefit from more nutritious and healthier food. And here is just summarized to just drive this home also a bit clear how many functions or services we find in the soil that are very beneficial to us humans. So, so we have first the soil organic matter here, the soil basically in the middle. We talked about biodiversity and soil formation a bit, soil structure, nutrient cycling, and also water cycling. That is all connected and it's very important for the health of the soil. But all these basically support a series of services for humans that are usually categorized in 3 different fields. These are provisioning, regulating, and cultural ecosystem services. I just name a few here. For the provisioning, we talked quite a bit. Basically, soil provides habitat for other organisms. And thereby increases biodiversity and also increases resilience for biodiversity. It has a filtration function that we get clean drinking water, and we just talked about food production. Regulating services, biological control is basically to keep parasitic species, so pest species at bay, for example. Climate control is very important because we have a huge carbon storage in the soil. I will talk about this in a minute a bit more. And then natural waste recycling. This is nutrient recycling again. Then we have cultural services. And you can think about this probably like gardening, for example, is a hobby where people work with soil, and it gives them relaxation, for example, by doing this. Is a hobby we are using soil for. So a lot of benefits in healthy soils. And then, of course, one of the questions, what can we do to reduce and combat climate change effects? And one thing every one of us can do, and can be also a school project, is to have a compost. And I will end this talk with a few thoughts on this, why this is beneficial. Because I think if we all do our bit, then we can actually make an impact, a quite significant impact. First of all, it's important to know that a lot of organic material ends up in landfills, where we also dispose of various other garbage we produce. But landfills are the third largest source of human-related methane, so green gas emissions in the US, for example, and it's pretty much similar all over the world. And the problem is that in a landfill, these organic materials are not properly recycled, so they're just sitting there and decaying, basically, and there they can contribute 20 times more and produce 20 times more of greenhouse emissions as composting. However, if we properly use and recycle this material per compost, then those emissions become actually negative. So there's quite an effect on avoiding the production of greenhouse gases. However, composting, similar to decomposition in natural soils, and compost, of course, uses, for example, earthworms to help with this composting, has the same benefits as this natural decomposition process. You have a higher nutrient content, you have a better water holding capacity because of this microclimatic soil structure, and you have then, if you use this compost, you distribute it again into fields, for example, you may help avoiding erosion and runoff systems. The one other thing I wanted to mention here is that I talked about synthetic nitrogen fertilizers. It is much better to have compost with this natural fertilization process than these synthetic nitrogen fertilizers, because they contribute basically to an overabundance of certain nutrients and also to nitrous oxide emissions that are also not positive for the health of the environment. And composting, the natural process, takes quite a lot of time, this decomposition, and really have this very nutritious topsoil layer in nature. It can take, depending on the region we are talking about almost 2,000 years to produce 6 inches, about 15 centimeters of topsoil. But with compost, we can actually speed up this process. We can help producing actually this very rich soil. And I had also mentioned already that soil is a carbon binder. It's a carbon sink. So a lot of carbon dioxide can be bound in soil. So here's a number estimated that the world soil holds about 1.5 trillion tons of carbon in the form of organic matter. And here it comes again, this healthy soil versus unhealthy soils. That we have degraded soils that actually release carbon into the atmosphere. However, if we distribute this topsoil compost layer, we can, and thereby improve the health of the habitat, we can actually reverse this process. And composting, because it's so nutrient rich, contributes to plant growth, vegetation growth, and thereby we can bind even more carbon. So it's very important tool, actually. And in the bigger picture, I just wanted to summarize this, and with this we end the presentation, that degraded soils have been linked to the fall of civilizations because healthy soils mean basically food security. And you have a lot of security in a society. This simply helps society itself. You have more profitable farms. You have enhanced habitat for biodiversity for all these other creatures that live with us, and also more robust ecosystems that can deal better with changes and disturbances. And for a society, at a community level, it can create local jobs. We can talk about it and educate each other about environmental issues like we are doing here right now. And it may improve actually a feel of safety and community sense. It can contribute to healthy diets, and also that local communities may be empowered by the feeling or by these processes to contribute to composting and soil health. So a lot to think about in terms of what we can do and how important soils are. And I hope I have given you quite a good sense why soils and soil organisms are so important. And with this, I thank you for your attention, and I'm happy if you have any questions, and then we will see if we do the Kahoot or not, depending on how many people are here. Okay, thanks a lot.